A very good morning to one and all. I'm Dr. Neeta and I'm an assistant professor at the Department of Zoology, Sacred Heart College, Chalakudi, and I will be talking to you about genetics. Now it comes under the 5th semester of the degree course in Zoology, and we will be dealing with Unit 1 under the subject genetics, that is variations from Mendelian ratios. We will do this unit in two parts, that is two videos. In the first video, I'll tell you about Mendel and his work. This you would have done in your school or college, but this is just to refresh your memory. And also we will look into what dihybrid crosses, what monohybrid crosses. We look into a small problem related to Mendelian genetics. So in Unit 1, we look into the variations in Mendelian ratios, but I will start with Mendel and his work. You would have done this in school already or in the previous semester, but this is just to refresh your memory, because unless you understand Mendel and his ratios, you wouldn't understand variations in them. And we look into various examples under incomplete dominance, codominance, lethal genes, and pleiotropism. The learning outcomes or learning objectives of this unit are: you should understand Mendel and his theories based on the understanding of modern genetics. When Mendel proposed his theories, Mendel did not know about chromosomes or genes, and so you should understand Mendel's theory in the light of modern genetics. We'll move into then variations in Mendel's ratio, and finally you should be able to do some problems based on Mendelian ratios as well as variations in them. Now when we look into the introduction, it was William Bateson who coined the term genetics from the Greek term genesis, which means origin. Now, but it was Mendel who was the first to work on this and give basic principles behind inheritance, and that is why Johann Gregor Mendel is called the father of genetics. He was an Austrian monk and a school teacher, and because at his time they didn't know what genes or chromosomes were, according to Mendel, the unit of heredity were called factors. Mendel chose the plant, pea plant, Pisum sativum, and he chose it for the following reasons. He found that usually this plant is self-pollinating. Why? Because the reproductive elements, that is the stamen and the pistil, are enclosed by the petals. Also, he was able to obtain pure breeding varieties, that is homozygous varieties, and they grew easily in an experimental setup, and the generation time was also less. It was also very easy to produce hybrids because the stamens could be easily removed and pollen transferred into another plant. Hybrids were also found to be fertile, and he was able to find 7 contrasting characters which he had studied. Now let us look into the characters chosen by Mendel. He looked at flower color, flower position, seed color, seed shape, pod color, as well as stem length, and he found that in this one character always seemed to be dominant. That is, the first generation that was obtained by crossing the parental strains, one character seemed to be always expressed. For example, in flower color, it was the purple color that was being expressed in the first generation. So he said the purple color is dominant over white. Similarly, axial position in flower position is dominant over terminal. And green color in seed color is recessive, or the yellow color is dominant over green, and so on. Now, he did his work between 1864 and 1866, but this work went unrecognized. And later in the 1900s, it was De Vries and Correns who rediscovered Mendel's work. They did his work again and got similar results. Now there are certain terms that we will be repeating on and on, but I would like to make these terms clear to you. You would have an idea what they are, just so that we know clearly what these terms would be. Now when we write P, it would mean parental generation. These are generally pure breeding varieties, that is homozygous varieties, which have been inbred for many generations. F1 is the first filial generation, that is the progeny of the parents in the previous generation. Now F2 is the progeny of the F1 generation, which has been obtained after self-fertilization. As I told you, certain characters are expressed in the immediate generation or the F1 generation. These are called the dominant characters. Certain characters which are expressed only in the homozygous condition are called recessive characters. Now what is a phenotype? It is the physical appearance of the organism. So for example, if you take flower color, purple color is the phenotype of a particular plant, or white flowers are the phenotype of a particular plant. The genetic constitution of the individual makes its genotype. Now what is a hybrid? When you cross two individuals with two different characters, it results the organisms that are produced are called hybrids. When you're studying one pair of contrasting characters, so the cross between two individuals having these contrasting characters would lead to a monohybrid. Or when you look at two characters, say you look at the length of the plant, tall or short, and you look at the flower color. These are two different characters which again have contrasting characters, tall and short or purple and white. This would become a dihybrid cross. And you look at three different characters in two individuals, it would be trihybrid cross and so on. Now what is homozygous? When you have identical alleles that are present in both the gene loci for a particular trait. So I take, say for example, a trait such as tall, and if I look at its genotype, if both of them have the tall allele, then it is called homozygous. Or if you look at the short allele, if both the gene loci have the short allele, it would mean again it is homozygous. Now what is heterozygous? When the alleles present in the gene loci are different for that particular trait, it is called heterozygous. Now you know the different terms that we use. P means parental generation, F1 means first filial generation, the generation that you get when you cross the parents, and so on. Now let us look into a monohybrid cross that was done by Mendel. Look at this is the parental generation P when the tall pea plant was crossed with a small or short pea plant. Now we will use this letter capital T to denote the dominant allele, and you use the letter small t to denote the recessive allele. So when you cross a tall pea plant with a small pea plant or a short pea plant, we see that the first generation all the plants are tall. Their phenotype is tall. When you look at their genotype, it would be capital T, small t. Now Mendel, what he did, he self-pollinated these F1 generation and he got the F2 generation. He found in this F2 generation, for every short or small pea plant, there were 3 tall pea plants. That is, he got the ratio 3:1. The phenotypic ratio of 3:1. 3 tall and 1 short. This is an example of Mendel's monohybrid cross. So we see here the tall plants with capital T and the short dwarf plants with small t would give rise to the gametes capital T, this would have only one type of gamete, capital T, whereas the dwarf plant will have only one type of gamete, that is small t, and the F1 generation would have a genotype of capital T, small t, and a phenotype of tall. When these plants were self-fertilized, it gives rise to the F2 generation. Now the F2 generation, by the checkerboard method, we see capital T and small t are the gametes that are two different type of gametes produced by F1. F2, you see the phenotypic ratio to be 3:1 and the genotypic ratio to be 1:2:1. The same can be seen in black and white guinea pigs. Same way, the black and white guinea pigs, the F1 generation, all of them are black, which means black is dominant over the white color. And in the F2 generation, we get 3 black and 1 white guinea pig, which means the ratio is 3:1. Morgan, T.H. Morgan, he studied Drosophila and he was able to get a monohybrid cross between long-winged, you see long-winged Drosophila and vestigial winged or small-winged Drosophila. So he took the pure breeding variety of long-winged Drosophila, that is capital L, and when he crossed it with the dwarf, the vestigial winged Drosophila, he got the F1 generation. All of them were long-winged. That is, long wing is dominant over vestigial wing. In the F2 generation, he found the ratio to be 3:1. Long winged is to vestigial winged. And using the checkerboard method, here see the phenotypic ratio as 3:1 and the genotypic ratio as 1:2:1. And that is with the monohybrid cross, which agrees with Mendelian ratio of 3:1 or genotypic ratio of 1:2:1. Now based on this, Mendel proposed the law of segregation or law of purity of gametes. He said that in a heterozygote, the alleles, be it dominant or recessive, always remain together throughout without any contamination or mixing with each other. But during gametogenesis, they separate from each other so that each gamete will have only one allele, that is the dominant or the recessive allele. This happens during anaphase 1 stage of meiosis. As you know, meiosis happens during gametogenesis. It is a reductional division to ensure that only haploid number of gametes are formed. Now let me tell you what these homologues and heterologues are. This is an example of the karyotype of a human. Why do I know it is a human being's karyotype? Because there are 23 pairs of chromosomes. See the last pair, it is the X chromosome. So this is of a female human being. Now why do we arrange them in pairs? Now look at the first pair. The first pair is very similar in shape, structure, etc. These are called the homologues. When you look at, say, the third chromosome and the fourth chromosome, they look a little different in size. These are called the heterologues. So, each human being has 23 pairs of chromosomes or 23 heterologues. And each pair of chromosome is called a homologue. What happens during gametogenesis? We start with the mother cell. The mother cell has the homologues. So the homologues are always together. In this case, we have chosen a cell where it says capital A, small a. So it is a heterozygote. During the first step in meiosis, duplication happens. That is, each homologue duplicates, and this is called the tetrad stage because there are 4 strands of chromosomes. So what is happening? We have the duplication and the tetrad stage. During meiosis 1, these homologues separate, and during anaphase 1 they enter a different cell. And during meiosis 2, which is reductional division, each gamete gets only one pair of this homologue, so that after fertilization the male and the female gamete fuse together to form one pair. So you understand what happens during gametogenesis and what Mendel said. He said that these alleles always go together except during meiosis, when they separate and go into one gamete. So one gamete bears only one allele. Now we'll look at dihybrid cross. So in dihybrid cross we choose two characters. So in this case let us see seed shape and seed color. So we see round seed shape and yellow seed color is dominant over wrinkled seed shape and green seed color. So look at this. We see the parental generation. We take the parental generation and F1 generation. In F1 generation we get round yellow seeded pea plants, which means that round shape and yellow color is dominant over green color or wrinkled shape. Now when we do self-pollination of the F1 generation, we get the F2 generation. Now in this Punnett square method or the checkerboard method, we look at the gametes. We sort out the gametes on one side and the other gametes from the other plant on the other side. So we have capital R, capital Y as one gamete, capital R, small y as another gamete. The third type of gamete is small r, small y, and the fourth type of gamete is small r, capital Y. And we get a ratio of 9 yellow and round, 3 green and round, 3 wrinkled and yellow, and 1 wrinkled and green. So the ratio is 9:3:3:1. Based on this, Mendel proposed the law of independent assortment. That is, when you are studying two combinations of characters in the F2 generation, two new combinations, not the parental type. We started with the parent yellow round and green wrinkled. But we see in the F2 generation two new combinations, green round and yellow wrinkled. These were produced in the F2 generations. So the two pairs of factors or alleles, they behave independently of each other. And during gamete formation, the different pairs of factors, they segregate or assort independently of each other, so that new possible combinations are found in the progeny. Now we know that Mendel was successful in all his experiments. In all his experiments during the monohybrid cross, he got the ratio 3:1, or in the dihybrid cross, he got the ratio 9:3:1. Now, what was the reason behind Mendel's success? He concentrated only on one trait. Only later he combined the traits and studied. Also, he maintained a detailed statistical record of various progeny and their descendants. Now, when I tell you that he got the ratio 3:1, he didn't get 4 plants out of which 3 were tall and 1 was short. He got, say, 1,000 plants, out of which 750 were tall and the remaining were short. So he maintained a detailed statistical record, after which he was able to work on it and get his ratios. Also, he had a very critical mind which helped him in his analysis. But he was also lucky because he chose 7 traits which were found on 7 different chromosomes. Now, when we look into variations in Mendelian ratios, we will understand that there are certain traits which are found in the same chromosome, and this would lead to differences in the Mendelian ratios. He wouldn't have got 3:1 ratio if he had chosen traits that were found on the same chromosomes. Also, the plant he chose for his study. These are the reasons behind Mendel's success. This is just an example of the trihybrid cross, where you say take a tall pea plant which produces yellow seeds and round shaped seeds, and you cross it with a short pea plant with green and wrinkled seeds. You see the gametes in the parental generation have the genotype capital T, capital Y, capital R, and the recessive parent produces gametes of one type, that is small t, small y, and small r. The F1 generation, all the plants showed a phenotype of tall, yellow, and round, and their genotype was capital T, small t, capital Y, small y, capital R, small r. The gametes of these are of 4 different types. He found the F2 generation had a phenotypic ratio of 9:3:3:1 and a genotypic ratio of 27:9:3:9:3:3:1. I would like you to work this out. I worked it out for you here. This is how the checkerboard looks like. You can work it out yourself and you can find out whether the genotype and phenotype obtained is actually true. Now, what is a back cross? What is a test cross? When the F1 individuals are crossed with one of the parents from which they are derived, either dominant or recessive, it's called a back cross. When the F1 individuals are crossed with a dominant parent, there is only one type of individuals produced. But when the F1 individual is crossed with the homozygous recessive parent, there are 2 types of individuals formed, and this is called a test cross. Now, test cross is useful to identify the incompletely known genotype. Say, for example, you take the F1 individual, it is tall, but you do not know its genotype, and you want to know its genotype. What you do, you cross it with a double recessive parent. So, if the F1 individual had a genotype of capital T, small t, the offspring of this F1 when crossed with a homozygous recessive parent will yield 2 types of individuals, thus proving that this individual you started with, which has an incompletely known genotype, was actually capital T, small t. Now let us look into a problem. Now Mendel found that there are 3 traits in peas. So height of the plant, flower color, and pod shape. These are all determined by different genes. They assort independently. And if we take tall plants with violet flowers and inflated pods, and you cross it with dwarf plants with white flowers and constricted pods, you see that in the F1 generation, all of the plants are tall with violet flowers and inflated pods. Now, if these F1 plants are self-fertilized, the question is, what fraction of their offspring are expected to A. show all 3 dominant phenotypes, B. are tall with white flowers and constricted pods, C. they are heterozygous for all these 3 genes, and D. have at least one dominant allele of each gene in the genotype. So the questions are, show all the fraction of the offspring with all 3 dominant phenotypes, the fraction of offspring which is tall with white flowers and constricted pods, the fraction of offspring which are heterozygous for all the 3 genes and which have at least one dominant allele of each gene in the genotype. So I hope you will be able to solve this problem.